Okay, yeah, I think now it's already, yeah, almost right time. So we probably can already start. I welcome everybody at our last lecture of this course. It will be given by Professor Dieter Braun. You already all know him from his previous lecture. Today he will speak about more complex scene, more advanced scene, evolution of biomolecule precursors, and formation of biopolymers and first cells. So Dieter, please, the floor is yours.
Yes, thanks very much for everybody joining here. Second lecture. So last time we did a bit the basics. I now try to give you a glimpse of how things could come together. So although this is a model for DNA, RNA looks almost the same. It has a little bit more bent, tilted structure. The whole thing is how could such a molecule be used for the first steps of evolution. So we will go through these points. Basically, recap biological history. I think it wouldn't make sense to talk a bit more about chemical selection. So how do these molecules kind of self-select that they are more pure than expected? Biology is hallmarked for the fact that there's always very pure reactions, pure reagents. So we want to see that also in some preliminary sense, the origin of life. How can we copy sequences? You know, the natural thing is one sequence copies into the other, strand separate, goes back. How can that be implemented into number 4 geophysical settings, geophysical space? You know, we talk a lot, of course, on the early Earth because that's n=1's life we know, but hopefully, or likely, or we don't know exactly, other extraterrestrial planets could offer a similar thing or different, and then we can speculate whether some other molecules will make it on a different planet. And then how this sequence selection, sequence replication leads to evolution. So we've had this already before. So human going back for sponges, which are kind of also multicellular, built from eukaryotes. We now say about 2.5 billion years backwards in time. Bacteria, 3, 4, longer, hard to say. Most simple organisms we could think of, bag of molecules still, complex to assemble, get things together. And all of biology history kind of goes back to the genetic code, where quite a complex RNA machinery, the ribosome, which is fed by these tRNA molecules, which define the genetic code and which actually make proteins. So this is kind of where all the sequence back extrapolation stops, and we need to think about how such a molecule cloud could emerge by, we would say, the same mechanism which acted since then, which is Darwinian evolution, but not on cellular or already living sequence, where already living creatures evolve against already living creatures into always best adaptation. But here, molecules competing against other molecules, but kind of making such structures which make proteins alive and get to a setting to always more complexity. So we will think about what could be a physical setting and what could be a chemical implementation, and how would these things come together. One idea we will talk about is an air-water interface, where molecules accumulate at the interface, where you have water at the bottom, air on top. And it accumulates here, not because it's kind of amphiphilic molecule like a lipid, but it accumulates here because you have a temperature difference driving it to be there. And actually the larger molecules, and if they grow larger, will accumulate better at that stage. So it has some inherent push for complexity. Molecules we discuss will be RNA. I think, since this machinery is made out of RNA, why not try? And some people think it's, well, we keep on trying, and I hope we find new solutions to an old problem. So people, studied this since the 60s, and part of the papers we base what we do are actually from the beginning 70s, and we just have much better technique to approach it. And probably here and there we expand the parameter range a bit broader and find new things. So the hallmark here is this cyclic phosphate at the 5' end, and I introduced this molecule. So it's basically this tetrahedral structure here, and which is hanging. It's a bit hard to see if I oscillate this very fast at the ribose, and has the base over here. So this is a peculiar structure because, number one, we learn more and more. This is a recent unpublished result that we know how to polymerize this from these nucleotides. But that's only the first step. Those, if they still maintain that 2,3' cyclic, can come together to a template, kind of to the things it wants to copy, get on the other side, and have the same molecular group making the link and ligating this together. Kind of saying that this strand is copied into the linked strand up here. Two things need to meet. You know, the yellow and the green one needs to have the right sequence. So it's short enough. That's actually quite safe because any base mismatch would not make it attach. And number two, the ligation must take care that the last bases are also correct. This comes together and you have a copy. This is a rather slow process. We try to find conditions where this is getting faster. We get about 40% yield in a week. It's a bit sluggish, but that could be a way to go for Darwin evolution. Since this is sluggish, we want to test such replication settings here with faster mechanisms. I want to explore the physical side, assuming that at some point later down the road, we will find a faster reaction here and then can, you know, put things together. So one of the things we would long run aim for, of course, we'd love to get some structure like the bacteria up here. And as I said, you know, this is kind of a cross section here in blue from what we have up here, where we have a warm side, cold side. This is 10-degree difference, about 0.5 millimeter thick, where the water on the warm side always evaporates and recondenses on the cold side continuously by making bubbles. These bubbles grow by surface tension and grow so large that they plummet back into the solution. You know, that means that initially you make these bubbles, so this interface goes down and you have a very strong accumulation of molecules here. And as they accumulate and they get together, get more complex, they accumulate even better because this is basically a diffusion limited process. So you drive it into complexity. And the funny thing, just recently we saw that if you have a modern lipid, we yet have to, you know, organize a bit and try to get also older lipids. If you put that in here, they actually accumulate here, make tiny, tiny vesicles, larger vesicles. And what's the funny thing, at the same time, the DNA or RNA you put in there is also accumulating. And the movie you can see, and one of these air bubbles, you know, it's not only a large interface like here, but any air bubbles which you always have in microfluidics, always nasty to have them, and we always wanted to avoid them. Well, they actually turn out to be quite good, especially for this one. Bubbles seem to be a tad better than these large interfaces. So they accumulate now in this inner center. So you have to think that this accumulation site here is kind of at the center of this bubble because that's how it roundly touches the warm side. And what you'll see then in the movie is that it accumulates, we hopefully see it, accumulates material here, and you actually see how things make bubbles. Now, the interesting, important point I have to tell you here, you're not seeing the lipid molecule, but that's not what we stain fluorescently, but we stain the DNA. So what the process does, it accumulates the DNA about 35-fold and packages up into vesicles, which are actually very similar to what the cell looks like, or a protocell. So while we have devised this setting here to go for RNA, it seems to be also fit way down the road in evolution to make cellular structures. So the thing which emerges, and we have now a proposal written up with Hannes Mutschler, who's expert in RNA and ribozymes, and more the biological side, like pure systems, you know, how does machineries purified from modern cells operate, is that we would have air bubbles, they would have RNA, but the RNA here would hopefully replicate. Have the ribozymes or other more complex structure emerge, have the lipids together, pull it up into vesicles, and eventually even go towards modern systems. So all of that, and that's the important thing, without pipetting. Okay, so we have to, and that's a bit our specialty, which we were pushing, look out for physical non-equilibria to drive this, and not human non-equilibria of Eppendorf tubes and whatever things you mix and two things together, but rather device of non-equilibrium system which, you know, occur in nature and do this by themselves. Temperature is one driving source, but you know, who knows? Relative humidity oscillations, wet-dry cycles by humidity of day-night cycles or something like that could also be on the table. So where do we start? We start with these molecules and we want to go for a replication selection scenario. So any Darwinian evolution has an aspect that the molecules can replicate themselves, multiply, get more out of less, and would select. Now, the big question is if you have such an RNA molecule is what's the selection pressure? So what should it select against? Well, the most simple way to think about this is the ones who can replicate fastest by their sequence. And it doesn't have to be potentially build up a complex sequence, which has a very fancy structure. It's just that it, you know, can bind faster to some other competing templates, or it can have some other ways why the sequences, as they're shorter, for example, can replicate faster. They will, you know, multiply faster and therefore outcompete the others. One of the typical drawbacks you have in some of these scenarios is that short RNA strands replicate faster. If you do, for example, base-by-base replication, that's a big drawback. And therefore, you have a species where the shortest molecules run the fastest and dominate this population, and therefore will not go very far because you're kind of stuck in law of sequences, and Darwin evolution, for more complexity, you want to go for larger sequences. So the things we'd love to combine, let's see how well that works, is, you know, on the one hand side, we ribonucleotides, you can go for 4. You could make the argument that 2 might be more stable, but it might be also faster because the sequence space is smaller, therefore they find themselves faster. But as we now know, we have 4 bases, and by that we can adapt better to temperature because these GC pairs are very strongly binding for high temperatures, while you go for AU for low temperatures and have that balance possibility. But early in life, it might be difficult, right? If one of these 4 things are missing, for example, the C is missing, the G can't replicate anymore, and then eventually it will, you know, not make it, and you're stuck with AU pairs again. So for us, very often, we also always try to also look for actually 2 base sequences. It would mean that we could run life then only with 2 bases, less molecules, you know, more probable lower entropy state, higher entropy state. So would be a way to do it. We'd like to connect these with amino acids. With some success, let's see. It's a long story. We think because now biology, these molecules make these, you know, assemble them into proteins. Why not would it be great if these wouldn't have an early role early down the road? And all of that driven by physical forces, no pipetting, as I said, and the idea which people always thought about that, you know, if you think about the ocean, beaches, it will dry. That's kind of a large scale setting with the big disadvantage that if I'm here RNA molecule and by diffusion I go out there, you know, I'll never come back. You know, it's not like the boats are, you know, it will accumulate the molecules. No, it will just be washed away. It will never see another molecule again. So these large scale oceans are deserts. And I would even argue that likely also early Earth, it would be rather desert. Even in hydrothermal vents, it will be a difficult way, partially because this chemistry here needs dry states. For that, you need, if you're really down deep in the ocean, you have a hard time to make these drying reactions because pressure is so high, so you can't have gas bubbles. If you're a little bit closer, if you have hydrothermal vents near the Iceland, why not? Our idea is, you know, life is on micrometer scale. We have microscopes. We should go small scale. And why not do this microfluidic beach where it's in a closed thing, so the molecules essentially don't have to go away. They can be confined. Or, you know, you can have some inflow, outflow, but as you want. And this would be kind of in a rock bore. That would be a setting we'd like to to see, and that's what we do experimentally to figure out. Lucky enough, we got very good funding. You see, starting advanced grants. We also have a CRC, which is a collaborative research center in there and running again, actually, starting April. So we're actually looking out for PhD students at this point. And this actually also delivered quite interesting biotech company, if you want to check out Nanotemper Tech. It's a biotech using thermophoresis, which we explored in the beginning. So chemical evolution. So first, more about the purification of this molecule. Well, one is building up structure. You want to have, without too much additional molecules, make this longer. What we do here is we go for high pH. That's a bit the specialty we're pushing. And it turns out that if you dry this in wet reaction, it's not doing anything. If you dry this, it will polymerize, but because the dry state, it will not have the capability to sequence replication. So what we do is we create just random sequences. The question here arises that what linkage do we have? At this point, our conditions is giving us about 50-50 chance to have the right linkage. That's the biological one, 3', 5'. And the wrong one, which is docking here at the 2', is 50% of cases. So now the problem is if we have a mixture of these, or we have too much of the 2, 5', making these double-stranded form is essentially quite more difficult. And people have argued this is the killer. And yes, I believe we need to improve that number. But yeah, is that 50% half glass, half full, or half empty? To be debated. The point here is that if these strands, and I just, mimic those are the bases, and this is kind of this cyclic ending, if they hydrolyze back, so if they actually break up again, the good thing is that we get, again, with high probability, these cyclic ends. And actually, we do have some evidence that this recycling is faster for the 2, 5' linkage. So it has a tendency, if we run it through this recycling phase, that it should lean a little bit more towards the 3, 5'. What for sure it does, and that's well documented, that if you have the 3, 5' and you have enough salts, but not so much, single, physiological conditions enough at cold temperatures, and this reaction runs up down to 4 degrees, you get these double-stranded formation, and there the 2, 5' linkage supposedly to be values range between 100 and 1,000, faster hydrolyzing. So the molecule, and that's this chemical selection I was referring to, if it manages to get the double-stranded form, will actually save itself and not be degraded so fast. So why this is interesting, it's interesting because it implies a size selection. So if these molecules manage to be long enough, might be 8 mers, 6 mers, 10 mers, depending on temperature, salt, and so on, to get these double-stranded form, it can survive much longer. So what it needs to do in doing that is actually get the same number of the both bases, otherwise it can't form it, right? You need, say, GC or AU, the same amount, but that's actually already selecting for these two together. So only those molecules will survive who have that one-to-one mixture. So it has some interesting mechanism built in without much additional things. It's just built into the single RNA molecule that it can self-select. We just need to give it a good setting where this is actually operational. We hope that this would also give us some push for chirality, so that it's not only the 2, 5, 3, 5 linkage is a problem, it's also if you have a mixture of chirality, left, right-handed molecules, if they are mixed, they're also not able to make these double-stranded form, and again, you would select against it. So it's a bit the hope that we can devise a strategy where this actually self-purifies itself by just wanting to survive longer advanced hydrolysis against the attack of water. And, because you have that, you have 2 with complementary sequences, so you might talk about some rudimentary replication by that. it will never really, strand separate and really replicate by ligation. It will be not exponential, but it's, going a little bit in that direction. So the interesting thing as you do the backbone recycling, get about 80% back into the right active form. That's nice. But you might be out of luck and you end up having the phosphate either at this 2' end or 3' end and opened up. The problem with these species is they can't go back for polymerization. So they just fall out. The good thing, we never see, or at least to our detection limit, that this phosphate actually fully falls off. So we don't have to reattach it fully. We only need to recyclize it. So we need to remove the water molecule in here and get it back to the cyclic form. There are aggressive ways to do that. It's always aggressive ways in chemistry. One thing we would devise more is to have a triphosphate possibility. Why? You get this quite pure from phosphate at high temperatures, but and it helps a bit if you add actually ammonia to that. So Ram Krishnamurthy actually combines these and argues this makes a DAP molecule, and that could also help. So there are various ways of doing it, and one has to see, what's most probable to do it. So we see about 1% recycling if we only go for the trimetaphosphate, which might be an interesting way, because that's also a way to make this molecule in the first place. As seen here, so you get about, if you have no phosphate, you get the cyclic phosphate of 1%. Now, chemists are saying 1% is enough, you don't get enough molecules. True, but if this is, recycling, I just need to put in molecules which the accumulation machinery drops out and really loses, and you might need not to have these high yields in the beginning because you have a good recycling machinery. Let's see. These high pH values might also give you a new way or other ways to make up these molecules in the first place. Well, that's probably more left to the chemist in the field. Sorry. RNA oligomerization in the dry state. These are the data. These were initial data where we did a slow drying protocol. It was so slow that actually at the elevated pH here, we were keeping the molecules too long at elevated pH and therefore lost the molecule because the drying rate was so slow at 20 degrees. It took, more than a day to dry. And then we thought we need the temperature activation. That actually turned out not to be true. If you go for 4 degrees, you actually polymerizes almost as well. Could argue as the big advantage that you get a lot of these 2,3 cyclics left. And since then, we improved the protocol and get better ways to get a very good polymerization and keep the 2,3 cyclic. Still attached to it. So the temperature was not necessary for polymerization. There are other, cyclic 3,5' where you need to go for temperature for activation. Not the case here. That's actually quite good because we can really now go for reactions room temperature, where this double-stranded form actually is much more probable. How do we know about the linkage? On the one hand side, you can do protein digests, and in some applications, that's a good way to do it. In other cases, you can go for phosphorus NMR and learn how much of the linkage you have 2,3'. That's one way, but you need a lot of material, but it's doable. How do we know otherwise about our molecules? We do HPLC mass spectrometry. So we sort by length and then have the mass spec. So why I show this, it looks a bit flimsy, but it's actually, I think, much better than what state-of-the-art machine software normally does, because these are isotope peaks, 1 Dalton apart. And normally they're confined to one peak. So you lose all the information you have in this pattern, actually, and you just want to have the detection, you see the mass. We actually have built a homegrown software which is actually fitting the isotope to these peaks. And that's quite nice because we can do that even on Orbitrap data, which is more high-resolved mass spec, and even on FTICR, we are now adapting the software to do that. So we can be really rather sure about that we have the molecule. In this case, because we have this phosphate, the ionization in this AC electrospray ionization is actually quite quantitative, and we, independent of length of the molecule, get the same probability that it will ionize and go in the mass spec. For other molecules, that's a bit more tricky. So that, but for oligonucleotides, it's actually reasonably well that you get out concentrations from that protocol. So one methodology we can use this is to check right left-handed way of polymerization. So do we get a homochiral polymerization? I show preliminary results. This is just written up at this point. Hope I'm not punished by that. Keeps more interesting. So we do that by having a the normal version isotope labeled. Biologists use that a lot. It shouldn't react the reaction because isotopes are, you know, only very rare case where you have very strong. Anyway, I mean, if you ask the experts, they say it should be good. And you have the L version, the wrong chirality. And how do we distinguish that? Well, this is 5 Daltons longer. So if you have 1D in it, it shifts by 5. If you have 2D, then by 10 or by 15 DDD. And these are actually, again, you know, white, the measurements, colorful, our isotope fits on the measurements. That's why we can, for these trimers, can really distinguish. Do we have, you know, 1 wrong chirality and 2 wrong chirality, or the DDD version of it. So we can't know which of the sequences is. That's not what the mass spec will give you, but we can distinguish between homo and non-homo. And the idea and the hope then is that already here in this polymerization phase, we get the more clean molecules, because only the more clean molecules will be able to make then double strand. You know, that double strand you can go with LL or with DD, but if you have these mixed species in between, it will, you know, never come together. So better remove those already very early in the polymerization case. And some evidence of that shows that we are selecting for the homogeneous case if we have the base G. You know, that's where we are at this point. So you start with an enantiomeric excess. What does that mean? It's a relative ratio of D over L. So you have about 25% more of the D. And then you polymerize. And you have a look. If you look for all the polymers which you have, nothing is happening. We're disappointed by this because it just shows, okay, you get the same excess in all the polymers, which means that overall, all the polymers are polymerizing. The interesting thing comes here. If we look for the homogeneous only, so if you look only for that extreme masses, LLL or DDD, which you saw, then they actually go up to 60% by a factor of 2. You have more of either LLL or DDD. You know, even for long polymers, 4-5 mers, you get that. So that's quite interesting. It shows that apparently the arrangement of the molecules for the polymerization is so close and is so sensitive to the handedness of the molecule that it can keep itself directional. So these are molecules which are capable of hybridization. You know, we're not talking that this jumps to 100%. So one would have to make an argument this needs to be accumulated, then recycled again. The other molecules degrade fast and flush out, something like that. So slowly you get chemical selection of these chirality. It will likely be also very tough if it's, you know, between 0 and you want to get really by the small fluctuation, you have more molecule more than the other to get, you know, symmetry breaking of that. You know, in theory, it should be possible, but okay, chamber needs to be small, number of molecules, and so on. So that's a bit a tough probability. But you know, for the polymerization, this is okay, and that's spontaneous symmetry breaking you would only get if you have some selection mechanism which selects polymers more than monomers. So one of these bubbles, for example. So it goes also even for if you have it already quite pure, it still amplifies. So it's actually pushing itself very well to the extremes. So that has a good characteristics. So now we test that for G and C. C doesn't do that. So C is a much smaller molecule. Apparently it's not capable of stacking and doing that together, which makes sense, fine. It would then mean that it would rely on the base pairing is G, and that would then select it out by, again, double-strandedness. So this is not super precise as in an organic chemistry lab, but kind of how nature would do it, step by step. The second thing on that polymerization, the chemical selection, is a finding by Sarah Giroud, who's a postdoc in the lab. He was checking for GC polymerization, and mostly C showed the most dramatic results. We're now going for all the other bases too. And if you add these amino acids at this pH, where this amine group is known to have a pK, what we see is a strong enhancement of the polymerization. So I think we do know the mechanism behind, and we are writing this up, but try to prove that it might be general-based catalysis, which would be actually very nice. That it would mean that also already very early in the process of making RNA, it would be helped by amino acids. So the two molecules are not by chance later used and grabbed because they're anyway in high concentration around. That's a typical argument taken, that you have easy way to make amino acids as you make RNA too. So it's kind of a coincidence of both molecule classes. Here it would be a causation that the catalysis would link them together, and if they're together in the same place, that place would grow much better RNA and therefore drive the evolution and would be out-selected. So RNA would be fittest for places with amino acids. So then it would mean it's no lucky or no coincidental thing that RNA is amino acids around, because anyway around, because it's easy to make, but it's caused because if it's around, it replicates better, and therefore it makes sense to use that later for proteins, even for the point that the proteins would actually also make and optimize this process even more, right? I mean, right now we take about 2.5 or 5-fold more of these amino acids than the nucleotides. We dry it down and it does the job of enhancement. But of course, you can think of that already. Deep peptides or some very simple amino acid forms could boost this. This coevolution of RNA and peptides, and I had the trimetaphosphate in here, is an interesting way connected, and that's been shown by Olga paper in 1971, how actually trimetaphosphate can polymerize also the amino acids, but it could also make these linked molecules, very likely actually in the opposite direction here for the experts. We see 3 peaks of this molecule, so it could go on all the 3 points. This is just one starting point here. We do more experiments of this. We can actually get quite significant changes of the phosphorylation profile if we have amino acids in here. So there's an interesting thing going on. And that's the paper from 71 of Olga arguing that trimetaphosphate can cyclize the amino acids and polymerize from those by having phosphate at the amino acid end. And we do see peaks for example, here, diglycine with a phosphate. We see a triglycine. We also see other amino acids. We're exploring this. Right now, we're not so happy about the 60 degrees we see. There have been reports that this goes at lower temperature, which we couldn't confirm so far. So let's see how these two worlds come together. So when I said and told you that TMP is very nice to recycle the RNA molecule, it is also useful to enhance the polymerization of the peptides. So if those dipeptides, 2 amino acids together, could enhance the polymerization of RNA, this would be a way that these molecules could find such a setting. Again, this is kind of the peaks. It's a bit difficult to get the concentration of these. What I show here is mass spectrometry peaks of identified species of this mass. We would say, because we have the phosphate there, that the ionization should be stable as we saw it and know it through RNA and reliable, but it's a bit difficult to get these molecules. You have to make them, get the independent way of measure the concentration, and therefore inject it back into the mass spec to actually calibrate. So can be done, but depends on what your lab is capable of. These are things we try to explore, making the RNA nucleotides. The advancement here is we try to really get these Orbitrap high resolution mass spec, where you have the main mass here. This is 1 Dalton more, where you have 3 isotope peaks. These are overlap molecules. So we try to see what we can explore as we want to make these molecules bottom up. But there are a lot of questions of what orientation molecule you get, side products. We do get masses which match nicely with guanosine in this case, with quite good peaks counts. You get isoforms of 2 riboses together and these types. So you can have a look, but the orientation is difficult to ascertain if you can't go for NMR. So this is kind of a first screen, fast screen with mass spec we can do. And let's see, perhaps the conditions which we now know or learn that it's good for replication can be also used to make RNA molecules in the first step. So that was the chemistry selection story. So we try to convince you that this double-stranded form of RNA is kind of a good target because it selects molecules quite a bit before. And so it, while you might say, why do I make polymers where I have not yet the sequence defined, so I'm not yet making the sequence correct, I just make a bunch of random sequences. Well, what I do make, which is not random, is the correct molecule. So it will have the right chirality, hopefully, because that selection. It will have the right linkage. 2,5 is removed because it can't make this very stable, very, protective duplex. So the duplex here is not for copying, but it would have been chemically used for selecting my molecules. Now we want to go for the next step. So we have these polymers and want to see how can we use them for sequence selection. So we are exploring in simulators, planet simulators with the McMaster group. We tried a lot of experiments there, not really fully conclusive, but it's just trying around. One run looked quite interesting, but it was hard to reproduce and it was hard to fully understand what we're looking in here. So we kind of do these experiments more now in different ways in our lab and try to really understand this and fully reproduce it, basically. So these are given from the mass spec concentrations of these GC sequences. And in this particular run, which actually involved a long polymerization, many day-night cycles, but also quite harsh conditions, it looked quite interesting. And that's the pattern we'd like to see actually also in the future, that we are actually seeing here a pattern of concentrations. So what you see here is that the homogeneous GGGG and CCCC are at low concentration. And the mixed sequences, here, for example, it's 1C and 3G at high concentration. Now, if we're lucky, this would mean that this is higher concentration because you just have 4 sequences by which you can make that sequence. And that's why it's higher in concentration. So it could mean that we would have a balanced sequence polymerization of G and C, polymerizing very much the same. And we tried this afterwards a lot of times, and we are struggling to actually see this again. We just can't reproduce it. Happens, okay? But what we usually see now is that we have a lot more G than C in the sequence space, and that keeps things very difficult. And we have also some indication that actually proteins help us to balance it out. So the dream here would be, of course, that this would mean that although the sequences are very short, we talk about these double-strandedness, and therefore all the sequences are balanced out because only those which have double-stranded are actually surviving, and therefore you get actually these, not because you polymerized them homogeneously before, but you kind of pruned it and removed all the others so that only the 50-50 GC polymerization survived. Okay, so the idea is, whatever is here together survives, this one gets killed by hydrolysis. That would give us this pattern. And that, it's a bit the question, because you need high concentration, high salt to get, duplex, even with 4. That's a bit dubious, but, in this magic experiment, it worked, okay? If only we could understand it and reproduce it. So let's see, we will come back to that at some point, I think. But this looked very interesting. So we had cases where you have the same GC amount, which normally is never together, at such a very flat profile, where we actually follow the expectation just given by the probability how much sequences you have. So go in here. If you go for the 4 bases, there are also cases out of these measurements where these are actually quite nicely following the same pattern. So here the same thing can be done with 4 letters. Analysis 4 is still okay. 5 gets a bit tricky. So you have to really, tune the fitting routines of our programs to actually match the measured mass spectrum good enough. But it goes in the right direction. So this is the thing for the chemical self-selection we would like to see, because then we have a chance to get this, and we have a chance to then switch to sequence evolution, because things are actually capable of making a double-stranded form. Then we would love to see a template, 2 of these guys sitting on it, short ones, long ones, depending on condition, and they actually would ligate together and therefore copy the sequence. So this is what's termed a ligation chain reaction. So you ligate these things together. And of course, then the idea is that you would separate the strands and redo the binding, separation, binding, and therefore would make an exponential replication. So in computer models, this should work. And fingers crossed, that's one of the major targets in the lab we do right now to get this going. Because as you'll see in the next slide, we have these 2, 3 prime, sluggish and slow as they are, doing this reaction, and we need to couple this with a strand separation. I think we have a good chance because our reaction doesn't need salt specifically, so no magnesium and no things which make RNA really stick very tight. So we are able to separate the strands. So I think we're in a good position to show that. And then let's see, then we have different sequences and have a look how that evolves. We'll show you a little bit example of such a sequence selection later in the talk, where we sped things up by making the reaction faster with the protein, with the idea that we focus on, can we strand separate? Can we see what selection means actually? But we kind of throw out of the window, the prebiotic reaction. So we use a RNA molecule, which is, a protein, which is making the linkage, of course, kind of artificially fast. So these are things we'd like to see. What do these sequences look like, and what conditions can be used to make it? But first, for Adriana Serrao, she's defending her thesis in 3 weeks, looking for a follow-up PhD. Anyway, she looked for these ligation experiments and put these together. So what you have is gray, the template here, and these are the 2 strands, which we artificially made with some overhangs that you can nicely see them on a gel. And then you look, and after a week, you get a reasonable band. You can analyze this also on the mass spec and the HPLC, and the usual pattern emerges that at really low temperatures, the yield is not so good. So it seems to need a little bit of temperature to rise. If you have too high temperature, especially if you go at the same time with high pH, then you lose the whole backbone. Has to do, I think, in the meanwhile, a lot that we here have to put in a little bit of magnesium, which helps, but it's a dangerous agent, can lead to hydrolysis at elevated pH and temperature. But otherwise, it's actually reasonably well behaved. I mean, the experts here would see that it's not showing hydrolysis patterns in this. So we think it's not too bad. And we have, in the meanwhile, looked more close for how precise it is. And that paper is just came out in JACS some 3-4 weeks ago. I can read it in detail, what we did. This evaluation of such gels where we look out for things which are actually would be correct. So this would be the correct species. And if we have actually a G in here, it tends to have making some nasty things. So G is kind of the aggressive base, want to attract other things which should not match. So we see some other bands, but it depends on what sequence you have. Here we have an AG sequence, so that's okay. But if you, for example, go for AU sequences, these is much nicer and you actually get quite precise copying. All the details are in that paper. What you also get, and that's quite useful to build up structure, you remember that ribosome ball. If you have one primer not present, it actually makes a loop closing reaction, especially if you have here a complementary base like this G and this G and this C coming together and have another G, kind of this GC is strongly binding. You get actually yields of this much faster, much larger than the replication reaction. That's quite interesting because it means that your RNA structures you have can be used for building quite a complex structure. So part of it would be used to copy it, and part of it with this loop closing would be once you copied it to wrap it up to pure, how should I say, to make it into a riped state. Like proteins, you typically do post-translational here and there, some addition. So, and that could lead, from short strands, you link it up to make tRNA for translation and similar things. So the setting would be quite useful. We like it because, the conditions are very mild. So we should be able to strand separate these. But it is a bit too slow. If we put everything together, we have analysis of the hydrolysis of this. We get the ligation chain reaction in 2 weeks with a factor of 1.8 in the computer simulation. I think we should do it, but we'd like to make this faster. Still, it would be first time you could show a prebiotic ligation chain reaction. So the idea would be you have a template, you bring in the strands, the matching one comes down, they ligate in one step. So that's quite interesting. So the copying here would be length independent if the yield would be high. So it would be actually a quite good replication machinery if you could get it going. Need to separate the strands again. And as you bind it again, of course, you have to take care that these strands are not binding back to this. I think this is not a real problem because in all of these configurations, you have a lot more concentration of these primers. They bind, and because they're long enough, they don't unbind so fast. So the probability that you really get the full strand displacement of this back to this is not as high if you go for these ligation reactions as if you would go base by base. But that's to be seen experimentally. Let's see whether we can make it. And then you would, double it up, get 2, 4 exponential growth. That's the dream of prebiotic experimentation. So the prebiotic chemistry is sluggish, and therefore it is useful to, if we want to understand what the system does, just have a look what we do get. And here we did experiments where we consistently used fast proteins to run the reaction. I told you this ligation needs best case 40%, 7 days. If you have a ligase protein, which is optimized by evolution for the job for repairing DNA, so we switch here for DNA. The second thing, we think the base pairing characteristic is similar, rather similar DNA and RNA. Sometimes it makes life easier to run DNA because these proteins are easier to get for DNA. This does the ligation basically within 30 seconds, 100%, super precise. So you can imagine, what's that factor 10,000 faster experiment? It gives you much more possibilities to go over time, evolutionary time, and have a look what the reactions is doing. So the thing Toyabe was focusing here, is now professor back in Japan after the postdoc with us, was the following idea. So we don't go for full random sequences. Back then, we didn't have the sequencing capabilities, but we go for sequences A, B, C, and the complement. Okay, so here it's A/B/C/ and A, B, C. For the following idea, which Church had, was to think about if we keep this under conditions where they're just about to bind, which makes sense, right? We were polymerizing RNA just to the point that it makes it double-stranded. So it can just bind, and then this reaction will be slow because A and B just make it onto the template. Whereas if the history of the system has provided already one ligation in the past, for example, a BC ligation, this is very strongly binding to it because it's twice the length, and then A is easily coming to it. And it turned out that especially in this competition between these two reactions, this one is 40 times faster than this ligation reaction. So this makes the system, depending on the history of the ligation. So if it has a history that it had made already this BC dimers, so if we have some leftovers from some older times, it will actually, although the system can go in all kind of directions seen in this diagram, making ligation chain reactions, it will go in the direction where the old sequences steer it. So we would argue that's a symmetry breaking. It is, number one, a spontaneous symmetry breaking, but it can react to the memory of the past. And it has some very peculiar, interesting selection pressures, which are very simple to implement and very similar to hypercycles, which Manfred Eigen argued in the 70s would be interesting. We can argue it's actually more robust because this replicates all the time, and the hypercycles always needed proteins to operate, and it's very complicated setup, and it's hard to imagine how that goes prebiotically. So ligation could be interesting for symmetry breaking of sequence space. And I'll skip the details there. Please read the paper if you're interested. Second paper where we wanted to see what these ligation reactions are doing was this, about 3 years ago, where we start with random sequences. So we just give it 12 mirrors, random sequences. It turns out we actually had to go for 2 bases because the 4 bases were not fast enough because of sequence space. So those are actually, you would argue, why should they come together in the first place? Because they just find its partner and that's the end of the story. I'll get to that because this thing here gets assembled much faster because the sequence space is smaller. So there's a peculiar thing, again, how the fast and slow sequences, and that actually leads to a very complex evolutionary cascade evolving from these random sequences where only the sequences which are really fast in finding each other can actually make longer molecules. So number one, it has an inherent tendency to make longer molecules. You see that here, and we'll see that in a second, but that's sequence encoded. So the sequences and the mixture of the sequences are kind of the target of selection. So you don't have to make fancy sequences which fold itself, get a ribosome or anything. It has to match the environment. It's the evolution of the fittest, right? It's Darwin principle. And fittest mean I find a partner, right, for which these come together and they can link and ligate and therefore copy my sequence. So what we can do this, and this is now sequencing, we sequenced all of these strands and we could look at the short sequences, long sequences. Sequences are great if it works. Took us quite a while to get it going. So the initial pool is random. So if this is an axis where you have only T here sequences, only A sequences here, you get a Gaussian distribution. fine, right? So the oligo synthesizer you asked to make this for you was doing a good job apparently, and sequencing was, balanced enough. You did enough error filters to get this correct. If we have the first thing to get the 24 mer, we actually find a distribution which is double peaked. So what it means, we rather have a lot of T bases on the molecule, or a lot of A bases on the molecule, but not so much the mixture in between, or not so much where you have an A end and a T end. Well, the story is if you have that, it will just form a hairpin, and that will prevent it to bind to other sequences and therefore falls out of the gene pool at the first stage. Okay, so we get quite a strong sequence selection, which you could say, I need the hairpins for ribozymes. Well, fair enough. You can do that with loop closing and ligation down the road. So I wouldn't care so much about that. But you are actually selecting the sequences which are really good at replicating, which are really good in keeping the sequence information. And it just self-evolves. You just start from random sequences. So you get a network of genetic molecules out of the box. So that experiment is done, and that's the detail. Here we go on fast pace. Again, we have a ligase, which there's no problem with the high temperatures, so that makes life easy. So we can separate the strands. We can really do the dream experiment. If everything on prebiotic chemistry would be as good as we hoped for, we can just have a look what's going on. So this is dissociating very fast, ligating also very fast. We give it only basically 2 minutes, and then we go high temperature again. Why is that interesting? Well, that gave rise to, number one, the selection that these guys finding themselves will just not have the time, because you can do a back on the envelope calculation how fast they can find itself. So it needs an on rate of hybridization. How fast do RNA come together? And that gives you a time if you divide that by the concentration of the whole strands. But then you have to divide, here, multiply, because it's on the other side, how many of these you have. And how many you have is dependent on your sequence space size. So if these are 12, it's 2 to the power of 12 different combinations, and only the right ones, because temperature is matched. It might also have some mismatches. That's, it's back on the envelope calculation, gives you the right match together. So that needs 400 seconds to find each other. By the way, this is with 2 bases. If you have 4 bases, it's a factor 4 to the 12. You do the math. It's a lot longer. So going and starting with 4 bases in such a scheme will have some speed disadvantage. So simplicity gives you speed actually here. But if you have such a ligation site, which we want to have for the ligation here, you have 6 on the one hand side, which populate themselves independently from the other side. So you just can consider 1. So it's 2 to the power of 6, and that's 6 seconds. No problem to do it with these 120 seconds, because the ligase is actually anyway fast to dock and then do it, but it needs the hybridization. And that's what we are seeing actually. That's why it actually wants to make long sequences, because the other one is kinetically disfavored. So that's a funny thing. If you apply a temperature different, cycling or a wet dry or day-night, it will create an out of equilibrium hybridization system. So the things will be driven, and will be driven in sequence space by these timing arguments. And you will push the system to become fast. You push the sequences to be fast finders. And these are now the numbers for the 4 bases, 19 days, right? That's at high concentration. So if you think about origin of life, life was circling, Earth was circling faster. You have a 6-hour day. Doesn't look likely. So arguments for, in this scheme, right, of things, for 4 bases, for 2 bases. And that's what we saw actually in experiment. We tried hard to get the 4-base system ligate, and you had to be really slow, and you see a fine band. And this is again an experiment with 2, which shows you how the cycling time, it's confirming these numbers. So if you give it too long time, they find each other, and therefore are not ligating, but because once these are bound, they have a hard time to outcompete these constructs. So our analysis here showed that if you go for longer time, you're not ligating, not because you give it enough time and therefore it should actually work, but you don't get much temperature cycles in the first place for the whole replication. So it is setting a time axis, the molecules you choose. Okay, we have a system where we do need that temperature here, explicit temperature, but that's fine because DNA can stand it. RNA is always problematic if you have too much magnesium, so we'll need to balance all that. And we probably rather do salt cycling instead of temperature cycling because it offers lower hydrolysis of the RNA. But where should this come from? So how would you, hands off, no pipetting, do that? And there comes geophysical space. So what would you imagine? Okay, we need the dry state. So really deep in the ocean might not be it. If you do this before the moon, might have had Earth. That was not so lucky. But it could also be that moon formation event actually brought us the rich chemistry in the first place. And really wait long enough, you'll get also a lot of water on it. So then you have to really hold on to one of these volcanic islands. That might have been the case, because otherwise you dilute yourself in the ocean. Of course, there are always people who are optimistic and think this is not just a dilute ocean, but full of molecules. Still, you need a dry reaction where you get that from. So I don't know. I take it that we rather think about something in between, think about something where volcanic islands are around that deliver automatically porous rock. If you ever been to Iceland, Hawaii, pick up any rock, you get a porous rock because the volcanic islands just do that. So that these things are just popping out of the ocean, and then you have these computer animated ways how they get the volcanic islands. Very dramatic. Well, if you go for Iceland for settings where there was a volcanic eruption 15 years ago, this is how they look like. So you still have very steam coming out. The steam can transport temperature and very well. You should know that from your computer because you have heat pipes. So you're cooling your processor by actually, it's probably not water, but a vapor, which is condensing and recondensing. You can run it on water pretty much quite well. So these are very good settings for setting up temperature gradients, making strong gradients. Well, how strong? Well, depends, right? And all of that is in, porous rocks, right? This is how, it looks like. Always fun to go there. So that's from family vacation some 8 years ago. Need to go back again. So one of the settings, one of the things we are pushing is thermophoresis. So if you make a crack out of this, and that's the latest design, it's transparent, but you can make very defined thickness in such a chamber where you make the thickness defined. It's a little bit thinner for the crack we showed you before. It's about 150 micrometer to run optimally for small molecules in water. And that's a combination which need to balance this convection, where the warm side moves up the water with the molecules. And as it moves up, the molecules go away to the cold side. As they turn around, those moved molecules to the cold side get assembled and accumulate down here. So this is an experimental demonstration by fluorescence, 20-mer DNA. We do know that RNA accumulates very much the same because it's all dominated by this, the charge. Nowadays, we cool this by freezing it to minus 80 degrees. So within seconds, it freezes, and then we can open it up and take sections of such a thermophoretic trap. So this simulates a rock crack. And Christoph Mass was really, he's a senior postdoc, emergent. He's now habilitating. Hopefully gets towards professorship. Let's see. Looked here, for example, for salt leaching basalt. And with Thomas Matreux is just finishing the PhD, they showed that at the bottom, magnesium accumulates much better than sodium. And there are some classes of ribozymes, like a sun-Y ribozymes, who needs this more magnesium than sodium in this setting. And therefore, we can show that this is much more reactive in here. And if you checked out the Nature paper of Christoph and me 3 weeks ago, 2 weeks ago, it also used this system to accumulate amino acids and see how that selects molecule classes and then boost prebiotic reactions. So this is one way where we can really select and get the molecules together. The other, if we now want to talk less about how to make these molecules, but more about how then we replicate them, we might look for some of these air bubble, gas bubbles in such a system, so it accumulates long-term with the thermophoresis, and we are seeing that the molecules I showed you in this lecture are actually always consistently, and I think it's not, well, that's to be discussed, right? It's not a coincidence. They are always the best accumulating molecules: trimetaphosphate, cyclic phosphates, and the amino acids which are accumulating, which are best helping RNA to make it, like valine. They are the best thermophoretic molecules for accumulation. So at least we're not creating a conflict. So what we are saying, if you would calculate the probabilities of all that, that would be quite significant boost for your probability to get things together if your mechanism of accumulation is actually exactly selecting and therefore purifying your molecules you are interested to make your RNA world of. Okay, so we go for now, for what follows, more towards these air-water interfaces, but keep in mind, in one of these thermophoretic traps here, all you need is to have an air bubble down here, and then you combine both things in one go. That's for sure one experiment we're pushing. And what such an air bubble gives you, it's already discussed before, is that the capillary flow of evaporating water accumulates the molecules in here, and it accumulates it always better if diffusion coefficient is slow. So here thermophoresis is not playing a role at all. It's directly the size of the molecule. So basically, exponentially, the concentration is exponential function of the radius of the molecule. That's your accumulation. So it's exponential. It's quite a stiff growing accumulation if you grow the size of the molecule. So once you have a specific size, it's actually very difficult to get away from this interface, and show you an example of that. One example was also already the vesicles. If the vesicles, are forming and fusing and get together, they're just stuck there because this is creating about 10 micrometer per second water flow, which then evaporates into gas, and diffusion prevents you to get from that. And that can be checked experimentally in the same chambers you saw before. So we make a temperature difference and have a look. So this is kind of time point 0. So this is some residual fluorescence from here, DNA sequence, and it might be just the optical effect of the interface and refractive index change things. We never really could figure it out. It could also be that DNA already has some tendency to be at the interface. But what is then going on is at the real interface, where this comes together, you get a very pronounced high line of fluorescence at the contact line, which builds up actually very fast. So that speed is actually quite a good argument for this, because opposed to the thermophoretic traps, which need a bit more time, more on the hour time scale, this runs on the minute time scale. And it follows, if we do a full analysis of this, we do typically COMSOL finite element modeling, gives you these solid lines, and the experiments, without adding magic, just putting in the numbers, we can actually simulate. So an engineer would say, oh, I understand my system, and that's a bit the motivation for that, like an engineer to understand these systems. So this is what happens in the water. So here you see what happens on the outside, and that's almost real time. So these bubbles show a quite prolific dynamic. So they grow, and then they vanish, and you think, what's going on? Well, they go typically back into the solution, and you look at this interface, you get higher volume, and then you make more bubbles, and it goes down again, goes higher and down. So what that gives you, and we'll get to that, is that if you have one of these big bubbles and they burst kind of at the bottom, you get very low salt concentration. So here you might have salt for having your pH correctly and making the strands come together. If these bubbles here, this is a monster bubble here, vanishes, you can actually monitor at the bottom the salt concentration by monitoring the double-stranded state of a, in this case, DNA molecule by a fluorescent thread pair. So, and then you actually see the strand separate within that seconds, the bubble burst, and you get low salt concentration. So this gives you a salt oscillation, and that's actually then is the way how you could think that these accumulated molecules get a shower of fresh water, you know, because water evaporates only without salt, and you get salt oscillation while you're accumulated at the interface. So only at the interface, these two things come together. And it was, of course, interesting to check out what you get out of this for replication reaction. We'll see that in a second. But what you also see here is that this interface, well, here is accumulating the material. You're not seeing it because not fluorescence here. You accumulate it, and then the thing goes down. So those are actually on the other side, on the dry side, warm side, are drying. So the molecules are accumulating, the water recedes, and the molecules dry away. So if you want to go drying for the polymerization, this also is an offering here. So you get the three things which we are searching now quite precisely. We want to have polymerization to make the RNA, so we need dry conditions. We want to have high salt conditions that the strands come together, okay, and that in water, so we can replicate. And number three, eventually, sometimes we want to have low salt conditions to make the strands separate. We are not wanting to do this so much with temperature. We did a lot with temperature in the past, but RNA is just quite sensitive to temperature. You want to go over the salt range. And I think this is one way to do it. We'll get to another way to do it towards the end of the lecture. Okay, so this is a polymerization result. This is already quite old. We could do it actually much better now, but it actually shows that the interface is actually competing quite well with the bulk dry. So if you dry that whole chamber, you get the results in gray. If you only evaporate what you have at the interface and then you remove the thing, you actually get better polymerization, which is indicative that the small volume which you have only at the interface is actually quite good in polymerizing. And apparently this accumulation of molecules drying is actually quite a good condition for polymerization. So that works, we get the molecules. What also works is that if these molecules would replicate towards more complexity, if they have a sequence which bind like this is a simulation, we make large gel-like particles so that they all come together, arrange itself, and then they are accumulating, of course, much better. I told you this accumulation here depends on the diffusion coefficient, which is linear to the radius. So if these molecules come together, they really accumulate very well. So we start here with these single molecules, which here are, you know, these strands. I think here we were still at 36 bases. By now we can reduce that in size down to 18 mers. So we come closer to the polymerization. You put that in and you heat it up. This is what you see. You see the accumulation at the interface. It recedes. And actually, when the droplet, which you're not seeing in fluorescence, comes down here, you get very sharp dynamics at the interface. And what comes off this interface is kind of this dragon of DNA. So we have fluorescent labels here. So we do know fluorescent labels is always attached to the DNA no matter what. And if we actually heat this up to higher temperatures, it will dissolve again. So you actually assemble here kind of a prebiotic DNA origami style, huge things, you know, these are 200 micrometer of large structures just by this effect of moving things to the interface. So that gives you an additional selection pressure, but it also keeps things moving into the double strand, make them move around. A lot of options you have from such a process. not the least that it will preserve it long-term, the sequence together. So you had a rough life early on, RNA world. If RNA world makes long enough strands to make such a structure, you can store your nice RNA away for a large time. If you come and it comes back here and you get these bubble drop, low salt, it will melt away again into longer structures. More towards autonomous evolution. So I told you we have this characteristic of accumulating and having the separation of the strands by the salt oscillation. Why not use the standard replication reaction biochemistry does, the PCR reaction? So we switch gears a little bit because here thing is that we have a high concentration of this called primer, which binds to our template once we heat it to quite high temperatures and then replicates base by base. This is only possible if you separate the strands. So specifically here, we wanted to look for two things. One of them is this system capable actually to strand separate even at temperatures where PCR would normally not strand separate, because we are helping here with the low salt conditions. And as we'll discuss, we're also helping with low pH conditions because we have CO2 in this chamber. And number two is, what are the sequences doing? Because the idea is actually that the evolution only happens at this interface, so we can feed it from below, quite long-term evolutionary dynamics. And it should have a sequence evolution in the process. So we start with short pieces of 50 mers with a melting temperature of 83 degrees. We are operating the chamber at a max temperature 10 degrees below, 14 degrees below. So they should actually never strand separate, right? And the usual problem with PCR reactions, if you keep it a little bit leeway, long strands will have trouble to replicate better than short strands because it's, a math thing, right? You need linear time to replicate it. So longer strands need more time. And then this comes into an exponential factor because you exponentially replicate your sequences. It keeps things very hard for long sequences to survive against short sequences. So we wanted to see whether this accumulation here is accumulating the molecules only there where you can strand separate, because only there you have the salt concentration changes and you have that pH changes, and separate it because we keep it a bit too cold. That is to be expected also for RNA, right? Because it is quite difficult for RNA to strand separate. And therefore, it would be interesting to see how the system can behave. But again, we simulated it with the DNA experiment. So what's happening? This is how it looks like. It's actually quite a nice movie. You see interesting aggregates forming, which are actually asking questions for what we've seen before. And then after a while, these are hours here, you get very strong fluorescence signal showing a very complex evolutionary dynamics. And you get more and more and more stuff, and you get more of these aggregates. You actually also get aggregates in the solution, which is quite interesting feature of the system, because it could even be that we have quite peculiar selection pressures going on, apart from the things we can see with sequencing in such a system. Okay, where are we heading? This reaction showed an interesting characteristics. It showed that only if we have CO2, we are replicating what we are thinking to replicate. So we gave it food of a 50 mer and a 77 mer. So if you do a thermocycler where the strands fully separate and have all the normal PCR, this is what comes out. If you put both together, it happens what I told you, that in competition, the short strands completely outcompete the long strands. Long strands have no chance. So evolution is kind of back on the survival of the shortest tyranny of the shortest, famously said by Spiegelman, and it has no way to go for long sequences. If we go now for our chamber, number one, we have negative control. If we don't give templates, nothing happens. These are now in the chamber. If we put both strands inside, also nothing happens. But if we have CO2 inside, we are just having that additional help of separating the strands, because as CO2 with this water, pure water, gives you a pH of 3 to 4. And that pH, when it drops down, is not only have low salt, but low pH. And that helps also to separate the strands because of charge effects of the DNA base. And what we're seeing is this thing up here. Initially, you thought, that's an artifact, it's just goo. Or, should we really look for that? It's very long, 1,000 base pairs. It's just a stupid thing. Don't bother about it. But it was there consistently, and it actually showed what we want to have for prebiotic evolution, that some real complexity comes up. So we sequenced it, and we actually knew from our analysis, it's a longer paper, arguing what are the real conditions for melting. So it depends on the magnesium concentration. That's the salt concentration by the salt dependence, but then also the pH, and you are down here if both comes together. So the falling of the droplet gives you low pH and low salt. That's where we are. But we could measure the full melting temperature of this. And this is the bulk. So up here in the bulk, you just have the strands together. They will never come open and they can never replicate together. So this can be actually now translated, and we'll get to that, into sequence space. So if we give you sequences, we can have a look, are we actually, in the sequence space, where does this melting curve operates if we allow it these low pH, low magnesium conditions, which we knew from the measurements. And what the experiment starts here, it starts down here because it has about 25 bases of GC and 25 bases of AT. That's those 50mers we want to replicate. But if we looked at the sequences, we saw some of these, but we also saw then replicates of it, which actually were getting longer and longer and longer. So we don't have the effect in the thermocycler where we get the shorter pieces, but we get the longer pieces together, likely, and that's what the sequence told us, by having, pieces come together and replicate again, and come together, replicate again, get longer and longer. But the funny thing here was that here the system's kind of stuck, because we knew that this, while initially by our initial sequence, it wants to go there, it has a very hard time to separate the strands, because our analysis of the melting temperatures told us that these are barely separating because they're already too long. And most of all, they have too much GC bases. I told you that GC is binding stronger than AT or AU for RNA case. So what we saw in our sequences were actually then longer sequences, and they're mostly up here. So what the system moved and evolved, because it was pushed by this capillary flow towards longer strands, it had to adapt the sequences to that. So it had to go for high AT count, because that allows the sequence to separate much better. And then it could go for longer strands. Now, the interesting thing, it could also have said, like the thermocycler, I don't care. I want to be 50 bases like down here. I'm happy with replicating that. No, the system, because it could only replicate at the interface, and because at the interface the molecules are accumulated, longer the better, it was pushed towards more length. I'd love to have that, because that's what, hands-off experiments gives me an evolution towards complexity. And what really was amazing for us, once we calculated what is to be expected from this melting curve into that sequence space, because this is the line we were expecting the strands to separate. So the evolution is a slave to strand separation conditions of our chamber. And to go for long length, because that was one physical force driving it, And the second physical force to be able to strand separation for replication, because otherwise you're stuck, you're not replicating, and the others are replicating next to you and go and get all the resources. That's exactly what the system did. It self-evolved for that setting. So what does that mean? That means that these interfaces look very interesting for evolution. I told you how at the end you can get vesicles, so you can really go all the way. In the meanwhile, with Hannes Mutscher, we looked for a system which uses a RNA-based ligation. That has a bit a tricky thing because you use this 200 base SUN-Y, which pops out of the blue. Of course, we hope that our ligation 2,3' will overcome that at some point. But it was an interesting experiment because we could find conditions where replication is done only by RNA. And you stick together from 3 parts on a template a hammerhead ribozyme. And because that hammerhead ribozyme needs to fold on itself to be functional, the function of a hammerhead ribozyme is to cut a piece of RNA. And for that, it has to remove the template. So we have the same strand separation problem, but this time quite more hard for RNA at much higher magnesium concentration. Number one, the accumulation mechanism at the interface was enhancing the concentration of magnesium at the interface against the bulk. That's actually quite a good reaction condition for these things. But then the oscillation of salt by things coming down was actually capable of separating the strands. And we actually saw that the substrate in blue, this fluorescent dye here, was cleaved away and therefore got away from this quenching dye on the other side. Only then you see it, only if the reaction happens. See it down here. Sorry. Show that we can run what we just saw with the PCR also with the ribozymes and RNA. So we are thinking in a good position to move the system to RNA in the future. That would mean we have the system going up here, have it get these things together. We try to actually make the SUN-Y by this. We almost managed, not fully. And so we start to ligate things together and you just bring things together. So these are molecular details, so I probably lose you in going to that. But the splint ligation with a 2,3' cyclic phosphate would be an interesting thing. I have not shown you for that PNAS where we looked for that random ligation. The sequence space image is actually quite informational rich. And we see how these complementary sequences are actually acting together and make a network of replication. So the idea is that we really can, explore this dynamics. So we either go for oligonucleotides which we cyclize, or we go even for the bases itself, make the polymerization, and go for replication. So why these settings are actually, could give us the full evolutionary trace to the point of making the first cells. I showed you how to make cells in the first place. What you can get out of these is also the accumulation of the biochemistry, and that's to be resubmitted soon. So this is biochemistry for the experts. They know this pure expression system. It is basically a mixture of proteins which is capable of copying from DNA, RNA, and making from that RNA protein. And once you make that, you choose a sequence which is fluorescence. So if you keep that at, mix it together, but you mix it together at too low concentration, nothing happens. If you put this in one of these stone-ferretic traps, which accumulates things, initially you thought that's crazy because why should be accumulation the same for all the compounds that you would, accumulate things threefold or fourfold, fivefold to get things together. But that's exactly what we're seeing. we are seeing that it accumulates the molecules homogeneously. The salts accumulate the same way, which is needed as different proteins. They accumulate very similarly, and it really is able to make a cell without a cell membrane. So up here you could feed the system, down here you accumulate it, but it's fully accessible. So you don't have the problem of the cell membrane, which is shielding things up and you have no connection to the outside. But you just are able to keep feeding it. This is how it looks in the experiment, and a lot of controls on, proteins, gels, and, these papers always go over 100 pages of supplementary material. It's quite painful, but fingers crossed we get through. Interesting, simple controls, just tilt the whole thing by 90 degrees, then you have no convection, no accumulation, and you don't get anything. So they're very strict controls here you can do. We can measure the concentration of the salt. So this is a salt accumulation, which we do need, and which is by the construction of that trapping mechanism, same thing. And we are starting to see also in these air bubbles accumulating things and make pure expression. So the next dream is, of course, to get this packaged up into vesicles and make synthetic cells. But yeah, let's have a look. But this is already, the bubbles are growing, so we try to optimize this. It actually looks quite good. It actually even shows that it goes better than the symphoretic trap or fast timescale and so on. If we switch it off, it just diffuses away. Okay, I'll go here a bit faster because yeah, we are looking for these interesting aggregates here, because those could be a way to move around genetic material. Also, the vesicles would likely be the first thing to do there. Again, we also see things that they're sedimenting. This we did it for a tRNA system here. It's now also published in PNS on using phase transitions. So these strands don't need to be so long to make actually solid state material. And this is how we started to make the vesicles. With Carsten Göpfrich, we analyzed these, and they're actually quite well-behaved, nice vesicles. Okay, so if we push for these porous things, I think we have a good way to go for these thermal cells. Nice feedable system for late development. And we always are obsessed about cell membranes. These are cells which have one large membrane over millimeter size. Biology is not always using these smaller cell compartments. So I think there are many ways of how to think about how these aggregates of proteins can run evolution before you make the hassle to go through making all those complex machineries which are putting things into the cell, and really go for kind of the spaceships of life of having the full membrane. Okay, that's all possible with funding. So I have Origins Cluster Initiative. We do a pre-application of that. We have the CRC alive again. So this was under reconstruction. We were successful. We have the next 4 years, and hopefully extensions of that in the future. And that's a new symbol. You can check out our homepage, molecularevolution.de. This is where we were competing to get the funding. So now we got it, and please apply for PhD positions. So up there, it's the stick for that. And if you want to look what's the latest and newest on Origin of Life, we always invite every 2 years for a 2-day meeting, very condensed format in Munich. Check out the website, LMUDE MOM 2024. We have a late abstract submission, can come with a poster, free of charge. Just stop by for 2 days of Origin of Life research. And that was the last slide. Thanks very much. Happy to get questions.
Yeah, thank you, Dieter, very much for this very exciting lecture. So this lecture is now open for questions. So please write your questions in chat. So, okay, yeah, maybe I start with a short question. At the beginning of the lecture, you showed this bubble which accumulate molecules, but then at the end, it looks like it's like exploding. What was actually happening?
We switched off the heating. Okay, so it's good you checked it out. So things were stuck very well, and we would not have seen it really. So what we, you would imagine that that day-night cycle could give you some oscillation of temperature. In this case, we were a bit extreme. We just switched it off, and what you saw is that they just detached and got away from it.
Okay.
So the
So it
idea,
happened so
yeah.
rapidly.
Well, look, this whole movie goes over many hours, and that's why. We had to slow it down at that point, otherwise you would not have seen it leaving. So, yeah, it just takes some hours to get everything together, process everything, and then get it together. Yeah.
Okay. Yeah, because yeah, exactly. That time could matter. How do you deal with this? So in your lab, you have quite limited amount of time, and for example, you said that 1 week, which is needed for reaction, it's too long. But in the nature, of course, yeah, the time scale could be much longer, and maybe even slow reaction could be important.
I mean, my usual answer to that is that in evolution, the fastest creature wins the game. So we are checking with our experiments, not only that we're not too much frustrated for these day or many days experiments, but we think if we find conditions where this is possible, those for sure will take over the evolution. So if you have these pores, if you have one of these pores where things are just good, and they replicate, on a fast time scale, wrap it up in vesicles, very fast time scale, and so on and so forth, it will the molecules will be consumed, they will come there and actually be burned away for evolution, and then evolution will spread from there. So it is not, how should I say, searching for just the best conditions because we want to get fast things, but because evolution would search for those fast conditions by itself, by the principle of evolution. It's this, survival of the fittest, and fittest means you replicate the fastest. So because then you proliferate the best. And of course, if we go for completely unrealistic conditions where you have a lot of pipetting, a lot of other things, then of course that probability distribution will kill you, because then people would say, yeah, probability is 0 for that very fast experiment. So that needs to balance out, of course.
Okay, yeah, sounds reasonable. So please ask your question, don't be shy. And yeah, I think I will continue. Basically, you start already with nucleotides. Did I understand correctly? So, but can you say at least a couple of words? How do you think those nucleotides are formed?
I mean, we did that one orbit trap experiment where we're seeing things, but and we make the argument you don't need to have too high yield of those because you purify them and accumulate them with either the physical traps or all that chemical selection. The idea, how I see it at this point, is that we want to get a good idea on the good replication mechanism, and then we want to go back again to see conditions to make those molecules under those conditions. So for now, people always argued for pH 7 and did all the experiments at pH 6 and 7. Now we see that replication is actually operating at 9, 10 quite well, and that's also motivated a bit by the Sweetwater pH on Iceland, and there are also phosphate lakes from Dave Kettling studied. They're all pH 9, 10. So it could be one should reiterate those experiments and think about what's possible at higher pH. So we just want to go a bit backwards in time. But then we're a very small lab with a few PhD. We can't go too much in the chemistry. So, but let's see. I would just not push so much for high yields to really stockpile nucleotides. and then hope that it will be eaten away by evolution down the road, and make quite some manual, processes in doing that. But rather think about perhaps we can really fuse those, replicating conditions and making it, but get away with have 1% yield but have it continuously. So let's see. It's ongoing challenge, I guess.
Okay. But could it be, for example, that it's not nucleotides polymerize, but only sugar polymerize, and then some additional groups are added, forming DNA later on? Does it make any sense, like sugar polymerization?
Well, I guess if you would have a 2,3' ribose, it would also polymerize. You don't have that selectivity and that information content, so you can't go for evolution. You can't, make those networks of interactions, because that is, for the templated ligation, the whole story is that I encode by my molecule which molecules come really close for that ligation. So if you would do that for ribose chains, you would need to dry it down, but then everybody links to everybody, and there's no complexity of that sequence network. You don't get. Sorry if I just go back. Hopefully it's fast enough. You don't get these too much. You don't get these networks of autocatalysis. People always discuss about autocatalytic networks. Kaufman did a lot of theory, but the argument we would push forward, and many hopefully I think agree, is that yes, the RNA is a very complicated molecule, but it's the simplest molecule for which you can make such networks from, right? So that autocatalytic network, which might be kind of the start of things going for prebiotic evolution, you need those complex molecules. So you need to accumulate them, wait for them, purify them, hopefully get them. But once you have them, evolution can start, and simpler molecules just don't do it. And people really checked for that. So you have to go extremely pH 14 conditions for Fomos reactions, and add whatever unprebiotic things into the solution to get to something a bit close to some autocatalytic, while here RNA gives it hopefully better, but with the help of protein it does, and let's optimize the ligation. You can really go for that direction.
Okay.
I don't know. People are too much in the astrophysics community to ask questions. I mean, come on, ask simple questions. That's fine.
Yeah, don't be shy.
I just fear that I was flying over your heads with what I told you, but I don't know.
Yeah, people just waiting the end of this lecture.
Well, that's the
series.
drawback of online things. People don't write down their questions, and then you should have written it already early in the chat. I could have reacted to that.
Maybe, one more question from my side. So,
thanks.
Instead of bubbles, would it work the same way? Oh, okay, we have one, but so I continue. Instead of bubble, if we would have a droplet, would it work in the same way?
Let's see whether
so
basically
could it,
if this would be water and this would be air,
like interface.
In a way, yes. In a way, yes.
A...
if this is kind of only a small bubble and then you have air around it, yes, you do need a bit that it spans over that part, right? And then you can still do it, sure. But if it's only sitting on one side, it might just evaporate. And that's actually something I hoped I had slides for here. But, with dry cycling, you could think about just droplets, dew droplets, which are condensing and evaporating. And So there might be even simpler things where you don't need the thermal gradients. Although, you know, my idea could be that you have a replication which goes, you know, wet-dry cycling over this 6-hour day and is happily, you know, evolving. But then, you know, structures like these will take over where evolution goes, you know, 1,000 times faster because you can do much faster replication cycles. And then life would have, you know, have prepared things already a little bit, and they still continue to prepare, make RNA. But then these are kind of the hot spots of evolution where really things go on and make vesicles and everything. And they're just using the rest as a feeding stock because they say, if I replicate my sequence every 6 hours, that's much too slow for me. I want to go for these megafluidic systems. So you know, that could be a hierarchy of settings.
Okay. So what do we have in chat now?
When do you do all these experiments, what gas conditions have you put in the chamber? In our case, we either make 1 atmosphere of CO2 and 1/10 of atmosphere of CO2. And, you know, they're both is reasonably fine because both are giving us this low pH setting. And that's, if you ask, you know, we put that in because we basically asked, you know, folks like Dimitar Saselov and other extraterrestrial planet guys, what would the early Earth atmosphere would have had? And then they would say, perhaps 0.1 or up to 10 bar CO2 was reasonable. So we put that in. But that's exactly quite interesting, I think, that, you know, gas composition of a planet, you know, which is more accessible for astrophysical experimentation, you know, has an influence, right? If we did these experiments without the CO2, we didn't get any replication. So things couple in unexpected ways, and we should use that as an example to do more and more complex experiments, and the more we learn about how these extraterrestrial planets look like.
So, yeah, I see. Those are all questions about the examination. So, yeah, you will have
You will get your
information.
certificates.
Yes. Information additional via emails. So don't worry.
Yeah, if some questions come up, please you know, get in touch with me, an email or whatever. Stop by if you're in Munich. Always happy to give a lab tour. And as I said, right, this mom. No. Why is that's a bit? Give me a second. I'll just reestablish. This
Sure.
mom molecular origins Munich is. I had it up actually long enough, I guess. Is a nice way to you know, have a bit way of looking into it and get some ideas. We do that, you know, once, but there are also NASA meetings, and there was a Harvard Origin of Life Initiative did a meeting. We have ETH has a center. I think they might do a meeting again. So just look a bit around. Things are evolving. We try to get the networks connect and going, and get things collaborate on the topic.
Okay. Yeah, we have one more question in the chat.
It is more possible to think of kind of live off solar systems. You mean outside our solar system? You know, hard to prove. You know, if I, you know, tend to see that the more we have these experiments, therefore in our brains we think that the probability kind of rises because we see more ideas how things could go around, and then you could imagine that the Earth-like setting might have happened before, and therefore, you know, probability is higher. But that's kind of only a theoretical extrapolation based on the experiments we see. You know, to get hard proof will be very hard, if impossible. I mean, people push, and I think that's very good. But it gives us more ideas whether, you know, other possibilities are out there, and then you can think about it. But that's that's always a very hard question. You know, can we get more possibilities? In my brain, you know, seeing how these steps really look like, I think, you know, having so many planets out there for sure, right? But that's a kind of a feeling and an extrapolation guess, nothing more. It could be on the other hand side, and there are also indications of that Earth was quite special, right? I mean, size of Earth is, you know, if you look for extraterrestrial planets, you know, Earth seems already a bit special, but it's a bit hard to detect. Then you had this moon forming impact, which seems to be quite very special. Then you had very good plate tectonics to keep us afloat over this billion of years. But then you say, oh, for origin of life, I probably don't need that. I might get it much faster. So always difficult to extrapolate from n=1, right? QLT, sorry, didn't get that.
Yeah, thank you, Dieter. Can you probably also shortly comment about the role of radiation? Do you think it can work as providing an equilibrium conditions, or is it really bad and can destroy everything?
You mean UV radiation, or you mean
Or any
real
radiation?
radiation?
Because I also heard about
Ionization.
nuclear
Yeah.
reactor as a driving mechanism, and yeah, things like this. And UV radiation probably, yeah, the same thing.
Well, show it, look into it. I mean, for UV, I mean, for other radioactive, well, why not explore it? And we'll learn by that. For UV radiation, some has been done for RNA. So RNA is interesting in a way that it has almost looked as the basis of RNA has been selected against surviving UV. It could be a late development because, you know, we had some, well, I mean, if you look into the selection pressure, the first genomes, you know, 1,000 bases or so would have under normal UV radiation, it's really tough. You know, you have to hide for UV. You'll get burned otherwise because, you know, one mutation is killing you already. And that's a very frequent, easy event. So it could be that that signature that RNA bases are good against UV. It could be a late development, you know, that you know, it could even be that the bases were switched towards those for some reason, and then the evolution of the genetic code only came about. But it could also be that early on UV selection was difficult. What I can't really see so much to see that the molecules have to really be exposed to UV, and then that's kind of necessary because then you have the trouble that if you have replication going and that UV thing in the same chamber or close by, it's a bit a risky business. If you have evolution of longer strands and then you kill the bases, if you do the math and you look for the radiation and really look for the numbers, it is quite tough for the RNA to do it. But
so it's better to be in the dark.
Likely, yes, likely. You know, that said, it could be that some precursors need the help of the UV. So if we can't manage to get RNA going without the UV, fine. I mean, then we have to think about compromise conditions. It's a bit difficult to say what that fingerprint that RNA is very UV stable. It tells us, I think it tells us that life might have evolved early on or at the beginning near the atmosphere, and it was exposed to light. So not so much the dark ocean floor, but rather on surface. But you know, all these discussions, you can always make counter arguments and specific things. But if you want to go for most simple conditions, those one would need to think about. But again, do the experiment, test it out, and then we'll have the parameters, everything together, and we can synthesize things. Synthesize in terms of finding conditions and actually do predictions, which are good conditions.
Thank you. So, yeah, people today really, relaxing.
Yeah.
So waiting forward for the examination or something. Okay. Yeah.
Okay.
Also, our time is coming to the end. Yeah. And thank you, Dieter, again very much for this exciting lecture and very interesting results.
Yeah. Thanks for setting up this lecture. I think it's important that we get everything together. Okay. Have a good one.
Yeah. Oh.
All the best.
Yeah.
No, it's just thank yous.
Yes. Yes.
It's the Abspann.
Okay, then have good day still and
have a good one.
goodbye.
Bye-bye. Have a good one.